To mainly lecture 6 from the summer, right? Remember, summer goes a lot faster than our semester, so some lectures, most of the lectures are the same, but some of the lectures, you're catching up, okay? So here we go with lecture 7 on chapter 19 that we're going to look at circulatory system, okay? So we are going to be looking at the blood vessels that are coming from the heart, going through all the body and then going back to the heart. So basically we know that we have two major types of the blood vessels in the closed circulation, and those are your arteries. And remember, those take the blood, direct the blood from the heart to the system. And then you have the veins that will bring the blood from the tissues after you had exchange of nutrients and waste products. So the veins will carry the blood from the tissues back to the heart. All right. Now, when you look at the arteries, we see that we have three types of arteries. So we have some very big ones, some medium-sized ones, and some tiny ones. All right. So the very, very big ones, those are the elastic arteries. The medium-sized ones are the muscular arteries. And then the tiny ones, you have arterioles, and those will feed into the very, very, very tiny ones that are the capillaries. So the capillaries, as we're going to see, those are vessels in a category like basically by themselves, because a lot of the features of capillaries is different than the features for the rest of those blood vessels. Then when we talk about veins, you're mainly going to talk about two types of veins. You're going to have the very, very tiny ones. They're coming from the tissues, so you call those venules. And then those are going to merge into bigger and bigger vessels, and then you call those veins. So you're talking about the very tiny ones that are venules, and then bigger ones that are veins, and those are just going to keep on increasing until you get to the very big vena cava that drops the blood back to the heart. Now, when you look at the total volume of blood that you have in the body in the closed circulation, you'll see that the distribution of that blood is not the same. It's not 50-50 between arteries and veins. So most of the blood, or about 60% or so of the blood, is really sitting on the veins, on the venous side. Okay, and then the rest is distributed along the arteries. Okay, and you see here that a lot of the percentage of the blood from the arteries is really part of the systemic circulation. So you have about 15% of the blood that goes to arteries and arterioles in the systemic circulation, and then about 20% of the blood that goes through the pulmonary circulation. And so then keep in mind here that the bulk of the blood, 60% or so, is really at the venous side. Okay. So these slides, these figures, I took from the worksheet that is found in the study session part of the Pearson site. Okay, so if you don't have access to that, I encourage you to watch it, because I find it very helpful, and the videos are easy to follow, and it's nice language, and it's a good review. Okay, so I do take several of the images from there instead of taking directly from the book. Okay. And if you don't have access, I will put the worksheet in the module, so you can just access from there. Okay. All right. So we know that your blood vessels with your closed circulation is basically bathing most of the cells in your body. Okay. So all of the circulation that is all the tissues in your body, most of the tissues in your body, they really have to receive nutrients from this closed circulation. But there are some tissues that do not really have blood vessels. Those are not a lot of those examples. Okay, so here we have them. These tissues, they are avascular. Right. Remember, if you took AMP1, the first type of tissue that we looked at was the epithelium, right? Was one of the first types, and we know that the epithelium, like your skin, the very top part, the epithelial layer, it doesn't have blood vessels. So those are the avascular tissues. So besides epithelium, you also have those tendons and ligaments. They also do not have blood vessels, right? And also cartilages. That those are, really don't really have blood vessels. So everything else, all the other tissues in your body do have blood vessels. So if you get your blood vessels and you stretch them out in series, you have a lot of miles of blood vessels. So they say you can wrap it around the world 2.5 times or so. Right? So that's a lot of blood vessels. If you connect also all the tiny capillaries and arterioles and all the other blood vessels. Okay. All right. So we've seen also that the areas of the body that do not directly have blood vessels, if they are considered alive, like live cells, they still have to get nutrients. They're coming from the blood. So usually those areas that are avascular, they are in very close proximity to a very vascularized tissue. And the fusion from that vascularized tissue, the fusion of the nutrients, is really what is keeping those cells alive. Also keep in mind that in order for cells in the body to be alive, they have to be bathed in fluids, mainly in interstitial fluid, right? Because if they are not, if they are just in contact with air, for example, with dry air, then those cells are not going to be alive. They're just going to be dead. Okay. So here is a very cool lesson map that I also just borrowed from that worksheet, right, from the Pearson worksheet. And the way that is organizing here the topic is that we have mainly 3 types of vessels to focus on. So the first one that we focus is really on the arteries and the different types of subcategories of the arteries. Then we are going to look at the capillaries, okay? And not only the subtypes of the capillaries, but also in this very interesting thing that is a capillary bed. So all of that exchange of nutrients and also of waste products, that's going to happen at the capillary bed. And that's where you're going to have the connection from the arteries to the veins. So here is, this is like a, what do you call it? Arteriole that is going to the little arteries. So here, this side is in the arterial side, this is the venous side. So in the capillary bed, you're going to have the arteries that get together with these little capillaries, and then they connect with the venules, and that goes to the veins. So all the exchange is going to be here, like between the fingers. Okay. So here is your capillary bed. Okay. So after looking at the capillaries, then we're going to look at the veins and the function of the veins as well as the structure, right, for the veins. Then we're going to take a brief look at these interesting things. There are anastomosis, okay, and what's the use of those in the first place, okay? All right, so here is a broader view looking at your book chapter map. Right, and I like this map because it's not only looking at structures, but it's also looking here at the physiological part of the circulation. Okay, this is the part that we have to use a bit more of brain power to understand what is happening. Okay, so we're going to look at different pressures and different types of flow And then we're going to look at what is happening here as the arteries get smaller and smaller. What kind of flow they have. Is it pulsating, or is it a continuous type of flow that you call laminar flow, right? We're going to look at resistance and what happens with changes in resistance that is directly related to changing the diameter of those arteries, right? So we're going to look at blood pressure and blood distribution, and then control of blood pressure. This part gets really, really interesting. I control the blood pressure, right? Then we're going to look at what is happening here with the capillary exchange and the laws that govern how you get that exchange in the first place. Now, keeping in mind that all the exchange that is going to happen is really going to happen at the capillaries. Okay, so you're not going to have exchange of stuff anywhere else but in the capillaries. Okay. All right. So here, again, I love that worksheet. So this is basically telling you all the 5 different types of blood vessels, the main 5 types of blood vessels. So here you go, coming out of the heart, you have the big elastic blood vessels, right? Big elastic arteries. And then you go to smaller arteries. And here arteries, you put elastic and muscular arteries in the same bucket, okay? Then you go to smaller arteries, and then you get to those arterioles, right? Very, very important for control of blood pressure. Let's keep that in the back of your head because that's a question that I will ask you, okay? All right, so here arterioles are control blood pressure. Then here we go to the capillaries, and that's capillary bed, and this is where all the exchange thing takes place. And then you go to the venues, and then you go to the veins, and then you get back to the heart. Okay. Oh, 1 second. You can drop 1 lowest grade by doing better in the cumulative final. All right. So coming back here to arteries and veins. So, looking at differently, do you have any more questions about the exam or anything else? No? Everyone's cool? All right. All right, so here, looking at basic structures of the blood vessels. So we have to keep in mind that all the blood vessels, most of the blood vessels, are going to have 3 basic layers, okay? So you're going to have the one that is very close to the blood, with very intimate relationship with the blood. You call that the tunica intima. Okay, so tunica is like a layer or a blanket, right? So tunica intima is that first part that is in contact with the blood. Now, what is making up the tunica intima? You basically have here 3 little levels at the tunica intima. So we have the first layer of cells that is endothelial cells, okay? And these are the same type of cells that cover all of these layers of the circulation, including the inner layer of the heart. Right, so we have these endothelial cells, this endothelium that is covering all of the circulation, and they are very thin, like fried egg type of cells, right? What do you call those types of cells? Do you remember? Oh, here we go. Audience participation part. Yeah. Yeah. Yes, it's squamous, so I don't need the thing for it. Ready? Okay. Oh, all right. You catch. Right. So squamous cells, right? So these are the squamous endothelial cells right there. So it's 1 layer, see, 1 layer of those cells, and they're flat, they're close to each other, okay? After this first layer that is direct contact with the blood, then you have these fibrous layers here. So you have this thing, these 2 layers that is part of this sub endothelial layer. Okay, so 2 parts that are in this thing. First part is called the basement membrane. Okay, so that is connecting your endothelial layer with the other layers. They're going to come deeper. And then you have this other layer here that is mainly made of loose connective tissue. Okay, so basement membrane is right there. And then you have a layer of loose connective tissue, and mainly you can have those areolar tissues, connective tissue here. Okay. So tunica intima, you have these layers, one endothelial, and then this fibrous layers, and then you have this very elastic layer, especially for the bigger arteries. Okay, and that one you call the internal type of elastic layer. Is an internal elastic sheath that you call it lamina. So this is very, very important for the bigger arteries, because those bigger arteries have to have a lot of elasticity, of course. They have to expand and then they have to contract. They have to coil back like a rubber band. Okay, so this layer is quite important for that. So that is what makes your first intimate layer. Okay, tunica intima. So after that, then you're going to have here the beefy part of your vessel. So the layer that is right in the middle, you call it the muscular layer, or you call it the tunica media. Okay, right in the middle. That is what is going to have your smooth muscle cells. So if you look here in this blood vessel, okay, you're going to see, of course, first you have here your first part here of your tunica intima, right? You see here that this elastic tissue is looking like Swiss cheese, right? But that's like the elastic tissue that is separating the tunica intima from the tunica media. And here is your tunica media. Look at the shape and the position here of your smooth muscle cells. Remember, your smooth muscle cells are not voluntary, right? They're involuntary. And they are laying on 90 degrees from the axis of your blood vessel. So they are mainly here like at a 90 degrees angle. Okay, they're laying down like that. Okay. Why? You should say why. Okay, so somebody from the back, I do want to use this name shot. Why are those cells laying down like this in this blood vessel? What is the function of that? Sorry, what was it? Contraction, like what do you call that? Contraction of that. Yeah, you have mainly these cells here that if they contract, what is going to happen? What is going to happen to the blood vessel? It gets smaller, right? So who said that in the back? I want to see if I can see you use this. Yeah. Oh, okay. Well, on this side too. What, this way or this way? I heard it coming from that side of it. It was you? Okay, so ready? Careful if you have a nut allergy, most of those have nuts. All right, watch your head, watch your head. All right. Oh, it's wrapped, so it's still safe. Okay. I'll get better at it. All right, so if they contract, you're going to squeeze the blood vessel. What is going to happen to your flow? Flow through the blood vessel, what's going to happen to it? It's going to go down. What is going to happen to your blood pressure? It's going to go up, right? And what is responsible for all this control in the diameter and flow and pressure? What's the name of that tunica? Media. All right, tunica media right there. So partially the tunica media, you still have another layer of Swiss cheese that is your elastic layer. Okay, so you have here external elastic lamina. Okay, because you have another one here is called internal. So this guy here further away from the blood, that one is the external elastic layer or lamina. Okay, so that makes up here your tunica media. There is only two sections, the beef part and the Swiss cheese. Sorry if I'm making you hungry, right? Okay, so after that, you're going to come to the outmost part of your blood vessel, and that is going to be your tunica externa. Okay, so this part here, basically it has a lot of fibers, because that's what's going to keep this blood vessel structure together. So we're going to have a lot of collagen fibers, okay? And you're going to find something really, really interesting, because you're going to see that this layer, the tunica media, you'll see here that it doesn't really have blood vessels right there. And then you have this elastic sheath here that also doesn't have blood vessels. And those cells are all alive and they need to get nutrients. And they are too far from the blood that is circulating inside of that vessel. So in order for you to bring the nutrition, bring the nutrients and take out the waste products from those cells, you need to have circulation in your blood vessels, right? And that circulation you're going to find here, that is a part of your tunica externa. So this is what you find here. You're going to find these little dots, those are actually arteries, right? And those little blue dots are veins, okay? So you have this tiny circulation that is bringing blood to your blood vessels, which is really a cool thing to think about, right? Then you call this circulation, you call those blood vessels, you call it a vasa vasorum, okay? So this is a circulation for your blood vessel. Besides that, you also have something else. You see the yellow dot right there? What do you think that is? All right, people from the back. With almonds. What is the yellow thing? Yeah, nerves. You did have another head. Yeah, you're right. Ready? Watch your head. Everybody's awake. I love this. Okay, ready? Oh, sorry. See, I tried to go now so far. Okay, can you throw it to the back? Oh, no, keep it. Here we go. Another one. I gotta get it better. Ready? 1, 2, 3. These have great aerodynamics, so we should. Ready? Ready? Whoa! Good catch. Okay. All right. So that was for the nerves. So you can send some information from your nervous system to your blood vessels. What kind of information would that be? Either contract or relax. We could also have some pain information coming from your blood vessels and right back. You should also send some information back. Okay. All right. So here is a slide telling you important things that you should absolutely know about those tunicas, right? And remember, if you have something that is red and bold and underlined, that's stuff that you absolutely should know. Right? All right. So here, let's keep on going here. And when we compare the two types of major vessels, right? You have your arteries and you have your veins. When you compare them, you're going to see that they all have those three basic tunicas, right? But now you're going to see that the veins are a bit different when you compare them to the arteries. Okay, first thing that is noticeable, although it's not very noticeable in this figure, is the size. My husband, he knows that I'm teaching. And I put my phone off, but he has a bypass on the phone. Okay, so I can't talk right now. Okay. So here, first difference. You look here at the diameter of the vein and you compare to the diameter of the artery, and usually the vein diameter is larger if you have comparable vessel size overall. So the diameter of the vein, the lumen, this is what you call the opening, the lumen of the veins those are larger than the lumen of arteries. Okay. Also, when you look at the muscle part of your tunica media, you're going to see that the veins do not have a lot of smooth muscle like you see with your arteries, right? So the veins, they are not really going to contract much, okay, in comparison to the arteries. That layer is really important in controlling blood pressure and overall flow, right? Now what's going to happen here, like a reason why the veins don't really need a lot of this tunica media, is because the difference in the blood pressure, when you compare what is happening with the artery to what is happening in the vein, you almost have no blood pressure going through the vein, very, very low blood pressure. So you're not really going to squeeze much of it to control systemic blood pressure, okay? And because you have such a low blood pressure going through the veins, what could happen is that the pressure is so low that it could backflow, back into the tissues, and you don't want that to happen, right? Because, for example, if you have veins that are coming from your legs, right, bringing the blood back in the direction of your heart, the pressure is so low that if the blood accumulates here, it could flow back, and that would cause edema. So you get very swollen legs, right? That's a problem, okay? In order to avoid the problem and keep the one direction of blood flow, the veins, they have these valves, right? And they serve the same purpose as the valves in your heart. Okay, so they keep the blood flowing in one direction, because when the blood tries to go back, then this is going to go right into the leaflets here of the valve, and is going to shut it close. Okay, so valves, you see them in the veins, but you do not see them in the arteries, because here you have plenty of pressure to keep the blood flowing in one direction. Okay, so that is a very, very important distinction from veins and arteries. So another thing that is really visible is the size of the tunica externa. Okay, so the veins have a larger, a thicker area here, a thicker tunica externa than in comparison with the arteries. Okay, so the tunica externa is really important for keeping the structure of that vein. Okay, and again, with the tunica externa, you also have the vasa vasorum, right? Because the veins, cells of the veins also need nutrition, right? And you also have those nerves going through there. Okay. So now here, when you look at the very, very tiny vessels, the capillaries, those are different. They have different structures than the rest of the vessels. Okay. So you see here, you basically have only the tunica intima, and you don't even have those elastic fibers or anything else. Okay, so you have this one layer of cells that is your endothelial layer, right? And after that, you only have this basement membrane that is keeping this endothelial layer together, but that's really all you have, because you really want to have opportunity for exchange of substances at this capillary bed level, right? So main differences of these types of veins, of vessels. So here you have a picture from your book showing an artery, and you see here the beefy part, right, the tunica media, okay, and how small this lumen is, right, in comparison to a vein here on the other side. Then you have a lot of tunica externa and a big lumen, right, and we don't really see much of a tunica media here. All right, but that's it's easy to recognize what is a vein and what is an artery. right? Another thing that is very interesting, veins are more to the surface of the body, right? If you look at your arm, if you have like fair skin or kind of see-through skin, you're going to see bluish blood vessels. So those are your veins, closer to the surface. So arteries are deeper. You don't really see your red arteries anywhere. And that can be a problem for you guys that are going into the healthcare system, right? Especially nursing, because sometimes you have to get samples of arterial blood. Ouch. They're not on the surface. I mean, if you have to go and give a sample of venous blood, it's relatively easy, although some of us tend to faint at the sight of a needle. Yeah, I mean, especially if you did not go through some, medical procedures throughout your life. Yeah, I'll tell you, things improve, right? But vein, already to get the vein is already complicated. Imagine to get the artery. Yeah, you gotta go deeper. You gotta know what you're doing, right? You gotta get a lot of practice. And I tell you, the nurses are the best at getting blood samples, right? If you try to get the doctors or the interns or the residents to get the blood, you suffer. Usually, you suffer. Nurses are really the best, right? If you know what I'm talking about. Okay. Oh, one thing, one observation. If you're going to get somebody's blood and you don't get it at the first try, don't dig through that. It is painful. Just take the needle out, get a fresh needle, start all over. Eventually you'll get it, or call somebody else. Okay, so, okay. All right, so, is it painful to get your blood drawn? Yes, it is. I think it is. You do have nerves that go through your blood vessels, and you do have valves in the veins. And sometimes when they put the needle through the valve, yeah, it hurts. Yeah. Okay. Just in case you never had that experience before. Okay. Because see the yellow things there, those are nerves. Okay. All right. So let's see what is happening here with the general structure and the blood flow. Okay, so first type of artery you're going to find very close to the heart, and example of this is your aorta. Is a very, very big blood vessel, like the size of a garden hose. Okay, big. So big elastic aorta. You also call those types of arteries, you call them conducting arteries, because they're going to get this flow of blood from the heart, and when they just receive that flow, they're going to expand a little bit. They have this elasticity, right? So that expansion is going to transform this mechanical energy into potential energy, and then the elasticity of those arteries that's going to push the blood back. It's going to bounce back. It's going to keep on pushing the blood. Okay, and that is important. That aspect, the characteristic of the elastic arteries, is really important to keep the blood flowing, right? Because if you're thinking about how your heart is pumping, after it pumps, it will relax. So if you don't have that further push in the blood, then you could have kind of like stopping your circulation when your heart relaxes, and we know that's not what happens. You get the blood always circulating, and this is really the job of your elastic arteries when they bounce back to keep that blood circulating. Okay, so here big ones are your elastic conducting arteries, and they do have a big lumen, but in comparison to the overall size of the artery, the lumen is not really that big in comparison. Okay, so so now you can identify the characteristic of your elastic arteries, because you are going to have this large size and very, very important elastic fibers. Now look at this. You're going not only to have the elastic fibers in the tunica intima and the tunica media here, but you're also going to have elastic fibers that are in your tunica media, like right there. You see the yellow things here? Very, very important in the tunica media. And this is another distinguishing characteristic from the other arteries, okay, from your elastic artery. Okay, bless you. All right. So after that, you're going to get bifurcation. You're going to have a branching of the elastic arteries, and they're going to go into this muscular type of arteries. So those are the arteries that get names. Okay, so they're bringing the blood to a certain organ, so and to tissues, mainly to organs. So those are the arteries that you're going to be blessed in the higher A&P courses to memorize their names. And no, you don't have to memorize the names here, but you should know basic things of what they're doing, right? So they're directing or they're distributing the blood to the organs, okay? So here with your muscular arteries, you are going to have a very pronounced tunica media, right? And still you're going to have relatively large arteries, like the size of a pencil down to the size of strings, okay? When you branch some more, you're going to get to these very important guys here, that they are the arterioles. So these guys just before your capillaries, that is the guy that is important for control of your blood pressure, right? So arterioles, you're going to have very prominent tunica media in comparison to the rest of the artery. And these guys are very, very, very tiny. They're the size of hair or smaller. Okay, so that's very, very small type of vessels. Okay. All right, so here is that idea on your function of your elastic arteries, as I just mentioned, with this idea of conducting the blood after you have a push of blood from the heart. All right, so here is your systole, and you're filling up the artery. Now, when you are going through diastole, this is when, remember, your aortic valve closes. So your blood is not going to go back to the heart, but now not only you have closing of the valve, but this is when you have the elastic rebouncing of your aorta that is going to help to push the blood into the direction of your system. Okay, so this elasticity is really what allows for continuous blood flow. Everybody cool with that? Yeah, all right. So here is more on your muscular arteries, so distributing arteries, right? So those are the ones that are going to bring blood to your organs, right? And in comparison, they have very prominent tunica media. Okay, so they are also pulsating. When you feel your muscular arteries, you're going to feel a pulse, right? Hopefully, right? So you have certain muscular arteries that are characteristic to give you a pulse. Okay. And that is really caused by the contraction and dilation of those arteries. Okay. All right, then you get to these guys here. They're very, very important, right? Your arterioles, and they are just before your capillaries. So the capillaries are the ones that do not really have tunica media or tunica externa. They only have the tunica intima, okay? So just before the capillaries, you have those arterioles, and those arterioles, they have very, very important muscles, smooth muscles associated with with areas that are controlling the transmission of the blood from the larger arteries to the capillaries. Okay. So what is happening here? You are going to have a lot of number of those arterioles in comparison to the other arteries. Okay, so most of your blood is going to be in the arterial circulation. Okay, When the arterioles contract those sphincters, that's when you can control the resistance of the blood flow. Okay, so basically the direction and the resistance and the overall flow is going to be controlled by those very much adjustable nozzles they're present at the arterioles. Okay. Besides that, remember that the tunica media of the arterioles, just before it gets to the capillary, the tunica media is very prominent in these arteries, meaning that when they contract, they can shift a whole volume of blood. Okay, they can shift from one area to another area. Okay, so it's very, very important for overall control of both flow and blood pressure. So arterioles are very, very important for that. Okay. So here is from your book showing the different comparisons, right, from the different types of arteries. And here is showing you in comparison how big each of these layers are. Okay, so if you look here at your elastic artery, you're going to have a lot of smooth muscle and you're going to have a lot of elastic tissue in comparison to the other arteries. But here, when you look here at your muscular arteries, you have also very prominent tunica media and structural, right, structural fibrous tissues. Then you compare with the arterioles, you have different type of distribution of those tissues. So be familiar with what they are and what their functions are. Okay. So here, let's see what is happening here to the arteriole. Okay, so this is arteriole. You have a lot of smooth muscle, right? And you have those smooth muscles that are serving as nozzles, adjustable nozzles. Okay, those sphincters that are between the arterioles and your capillaries. Okay, and a lot of times you're going to find that the arteriole is directly connecting one arteriole to a venule on the other side. Okay, so when you have this direct connection, that is also called an anastomosis. This is when you have this single type of arteriole that is going across. You call that a metarteriole. You're connecting directly arteriole to venule. There's not showing the venue here, but let's say, you know, the venue is like right there. So we draw the venue right here. Okay. All right, so we mentioned that. Okay. So here, from this metarteriole, you will also find here the sphincters that control the flow of blood to different capillary beds, okay, to the capillary beds here. Okay, so how is that happening? So look at this. You have here major arteriole. You have arteriole that is going to go through capillary bed, okay? So here at this junction of arteriole to capillary bed, you have what you call the terminal arteriole, okay? And here is where you're going to have a lot of these sphincters. They are connecting here to the capillary bed. If you contract those muscles, then you're going to slow down the flow of blood to the capillary bed. So here is showing that when the arterial muscle is contracting, this is when contracting the vessel, you call it vasoconstriction, you're not going to have as much blood flowing through that capillary bed. And sometimes you have a metarteriole that will go right in the middle and connect as a different path for the blood from the artery to the vein. And that's what you also call anastomosis, which I do have a picture here. Oh, finally. So let me see here. Okay. All right, so important things here. When your blood gets to the capillaries, that is the only site the only types of blood vessels that are going to allow exchange of stuff. What kind of stuff? Well, you have gases that go through by diffusion. You have water that mainly goes through osmosis, right? And you have your nutrients, and that goes also through diffusion, but not as easily as gases. Gases just go through the membranes like without any problem. Okay, thank goodness. So we can get oxygen in the tissues and get carbon dioxide out of there. Okay, so water, waste, and nutrients, everything is going to happen at the capillary level. Okay, not anywhere else in these blood vessels. You have to have capillaries to have exchange of stuff. Is that clear? Yeah, am I going to ask a question about that? Yes, all right. Okay, so here, let's see what a capillary looks like, general features of capillaries, okay? So here, remember, you only have this tunica intima, okay? So you have your one layer of your endothelial cells. Look at the size of this endothelial cell. See, the nucleus here is huge. Now you compare the size of that cell to the size of this thing here that is a red blood cell, right? So basically this capillary is so tiny that you can only get one red blood cell to go through at a time. And sometimes the red blood cell has to be squished and bent in order to go through, right? So very, very tiny. Now look here at the size of the cell, the size of the nucleus over here. You also see there are little vesicles here in this cell. So those are pinocytotic vesicles, okay? So those are basically vesicles that the cell is drinking stuff from the interstitial fluid, right? So you have this one endothelial layer, and between those cells, you have what is called intercellular space or cleft, okay? And these are the clefts, right? Cleft right here. And also, in order to keep these cells together, you're going to have tight junctions. You have tight, I always get those confused. Are those junctions? Yeah, those are tight junctions. Okay, they're going to keep it very, very tight together. Okay, you're going to keep the cells in a way that is not going to fall apart. All right, so tight junctions between the cells, and you have your clefts also between the cells. Like after this layer, you're going to have a very thin layer here of basement membrane that is also protecting and keeping the capillary together, right? Structure of the capillary. But remember, this basement membrane has to be very much porous, not porous, but thin. It has to be very thin, and it has to allow substances to pass through. Okay. Besides that, you're going to find some of those cells that are sitting on top of that basement membrane. Okay, so those cells here are actually stem cells. So those are called pericytes. They are very, very important to rebuild those capillaries in case you have capillary damage. Okay, and it happens. They're very thin and they're very fragile, and they can break. So pericytes just reconstitute the capillaries. Okay. Okay, so let's see here. I also mentioned which tissues do not have blood flow. Okay, straight from your book. All right. So types of capillaries. Okay, 3 types that you have to know. First one is the type we're just looking at. Okay, first one is called continuous capillaries. So those are the most popular. Okay, mainly a lot of tissues they have that. Like what kind of tissues? You have it in the skin, you have nervous system, all connective tissues, and you're looking at muscles. You're also going to find those continuous capillaries. So small clefts, tight junctions, and basement membrane. Okay, not a lot of space between those cells in your endothelial layer, right? So most common, all right. Now, after that, you're going to find these other types here. First thing you're going to notice is that the cells of your endothelial layer, they have these holes, but they're not big holes, they are tiny holes. So it's not like, they're not like doors, they're more like little windows, okay? So you call them fenestra. So you call those little holes on the cell, you call it fenestration, okay? And if you speak Italian, you know that finestra means window, right? So those little cells have windows that allow for more things to go through. Where are you going to find this type of capillaries? You're going to find in places that you need to have a lot of absorption or a lot of filtration. And here are your examples. You're going to find them in the kidneys, also in villi of your small intestines, and also in endocrine organs, because you're going to have to allow a lot of stuff to get out of that organ, secretion in bulk. So it helps if you have fenestration and those in the endothelial layer. Okay, when you look here at your basement membrane, it looks pretty much the same as your continuous type of capillary. Okay. So after that, you're going to have this other type of capillary here that it's kind of difficult to imagine how this thing is being kept together, because it looks like a lot of holes and empty spaces, and then you find some cells here and there, right? So what you have here, you have big windows, but then the windows are not enough, so you have like just bring most of the wall down, and you call those big holes, you call them sinuses, right? So you have windows, big ones, you have big sinuses, and those, you call those capillaries your sinusoids, okay? And you look here at the basement membrane, the cells that are making out the basement membrane, they have a lot of space. The cleft is huge. So you have a huge cleft within your endothelial cells, and then huge clefts here on the basement membrane. And really, it's like, how is that thing being kept together? Kind of amazing that it is together. And you're not going to find these capillaries in a lot of places. Okay, you're going to find basically in 3 places. You're going to find it in the liver, a very bloody type of organ. You're going to find it in the spleen and all other lymphoid organs, okay, like your lymph nodes. Okay, spleen, liver, and you're going to find it in the bone marrow. You're going to find it in the red bone marrow. Okay, so that's where you're going to find these huge spaces in the sinusoids capillaries, because you want a lot of stuff to be able to go through. And if you look here, the size of a red blood cell, even red blood cells can go through those spaces. And that will make sense if you look at your spleen, because the spleen is going to filter your blood, right? So your red blood cells will go through those capillaries and get filtered. And then if they are healthy to go back to the circulation, they'll go back. Otherwise, they'll be trapped in the spleen and destroyed. Okay, so they have to have enough space to be able to get out of there. Okay, so here are just slides showing the details of those capillaries that I just mentioned. Right? Let me see if I missed anything. So you have the cleft, endothelial cells, tight junctions. Okay. And here is a second type. Those are your fenestrated ones. Okay. And here is your sinusoid capillaries. And yes, I will ask you a question showing you one of those pictures and asking you what type of capillary that is. Right. And here, another question. All right. The following vessel types, which ones are responsible for the exchange of gases and nutrients within tissues, right? So this is a very easy question, especially now because it's so fresh. All right, does anyone need more time? Everybody answered? Oh, one more. Talking about exchange of stuff, gases and nutrients. Okay, I'm going to stop it. What is your answer? C. Okay. All right, so now you exchange this stuff, you left a lot of oxygen in the tissues, you're picking up carbon dioxide, and the blood is going to go to the venules. Okay, so this is after we dump the good stuff, pick up all the waste, and it's going to go to the veins. So the veins, as we mentioned, they're a lot thinner than the arteries, and mainly they don't have to be thick because they're not going to get a lot of blood pressure. Okay, they almost don't have any pressure. So if you compare the pressure in the venules with the pressure of the arterioles, you find this big drop in the blood pressure. So arterioles, they're gonna get, the blood's gonna get to the arterioles with a mean laminar flow of about 35 millimeters of mercury. What was the maximum blood pressure that you had as soon as the blood left the heart? 120. And then you got 120, and then you have a lower pressure that was 80. And then 120, 80, 120, and 80. And then you look at your muscular arteries and you feel the pulsation, right? You feel the pulsating. Now you look at your arterioles and you only find one type of blood pressure there. It's not pulsating anymore. It's laminar, it's continuous flow. But it's still some pressure, like 35 millimeters of mercury instead of 80 or 120, right? It's 35. Now you look after the capillary bed, when the blood gets back to the venule side, look at this, 16 millimeters of mercury. So that's really, really low in comparison to the arterial side, okay? So this is why your veins will need to have valves. Otherwise, the blood could just stay stuck somewhere and not really have a lot of pressure to get back to the heart. Okay. Now, what happens sometimes, you have certain veins, right, during some period of life, and the veins kind of get compressed a lot, right? Like, for example, when you're standing for a long period of time, right? Well, you're too young to go through this, but when you're older, when you're standing for a long period of time, if you sit down later at night and you put your feet up and you notice that your legs are a bit swollen, right? So you had some like muscular tension. If you're not moving around too much, you had, standing there, you had some muscle that was pressing on those veins. So the veins are having a harder time pumping up the blood if you're standing. Okay, if you're walking, it's different. If you're walking, your muscles are helping the veins to get the blood back to the heart. But if you're just standing and doing nothing, then you're going to have some fluid accumulation because the veins are kind of fighting with these muscles that are contracted. Okay. Now let's put it in even a more challenging situation. Let's say that you're pregnant. Now it gets interesting because now you're going to have this whole body of baby, especially during the third trimester, that is pressing everywhere in the organs, right? And babies move and they kick a lot. They love to use their organs like soccer balls. Oh, there goes, oh, hello. Yeah. And they kick your veins too. And they put so much pressure in there that the veins, they kind of like can't really get the blood back in a very effective way. So if you put a lot of pressure in those veins and you put a lot of blood here, the wall of the vein, the wall is not a lot of muscle in there, so it's going to dilate some, right? When it dilates, you see the flaps here of those valves. So here are the flaps, like there is the blood going this way, right? So when you are like pushing, like keeping the blood going in one direction, if your wall is extended, then the flaps are not really going to flap or anything, so that's a really, what do you call that, incompetent valve type of situation. And now you have some blood that backflows. And the vessel, the vein got extended and the blood is black flowing. And that's when you get varicose veins. And it happens To a lot of people who have children, to a lot of women who had children, yeah, you're going to end up with varicose veins. They're not pretty. And you're going to end up with a kind of a circulatory problem. And then the baby's born, and then you kind of put your circulation back in place, but everything is good. But you do have some situations like, yeah, later in life that you end up with varicose veins, and this is why. Okay. Okay, so here, this is your systemic circulation. This is the part of the systemic circulation. This closed because the blood is kept in the vessels, so it's closed. But besides that, when you have your capillary bed and all the exchange of stuff and fluid that's going on over here, not all the fluid that comes out of those capillaries, not all the fluid gets back to be deposited in the vein. Some of the fluid stays in the tissue, all right? The fluid that stays in the tissue has to come out of there, otherwise you're going to have swelling of the tissues, right? Otherwise you're going to have edema. So that's when you have these lymphatic vessels that come into action. So lymphatics, they're really open-ended little straws that are very, very close here, in very close contact with your capillary bed. And basically what they do is that they suck up the fluids by capillary action. Okay, so straws are there. Here's your closed circulation. Here's artery, here's vein, and here is the lymphatic, okay, lymphatic vessels. They are open-ended. They suck up that fluid, and by capillary action, it's not actually there. No, that's not what's happening. Okay? So just by capillary action, they also have valves to allow only one direction of flow. And what they are getting here is not blood, it's just the clear liquid that came out of the capillaries. Okay, so red blood cells are not going to be there, okay, unless you have damage. No, red blood cells are not going to go there. So only this fluid that gets here. So when the fluid gets picked up by those vessels, that's what you call the lymph. That goes into the lymphatics, and then it gets filtered, and eventually it gets dumped back in your venous circulation. Okay, it gets dumped here in your vena cava. Okay, so here is your closed circulation, and here is your lymphatic open circulation to pick up the fluid that gets out of there and does not get back in the venous side. All right, and this is the guy here showing this idea of anastomosis. All right, so basically this thing is a shunt, is an optional path that the blood can take, just in case the capillary bed is squished and blood can't really go through that side. You can have another pathway that is still going to get the blood circulating. So you're going to find this type of anastomosis in places where you have high pressure, like close to your joints, okay? Because when you press on something and the blood does not go through, you have this optional path, alternative route that the blood can take, okay? So those are arterioles. Okay, they're not capillaries, they're just arterioles that go across. Okay. All right, so here more about your anastomosis. Okay, so just interconnection of arterioles. You're going to find them in joints, and interesting here, you're going to find also in abdominal organs. Okay, and look at this, very important for the brain and for the heart. Why do you think that should be important for the heart? When the heart contracts, you can squish a lot of those capillary beds, right? So you must have an alternative route for the blood to go through, right? So this is anastomosis are very important for heart circulation. There's, yeah?
Can you explain why it's important for the brain? Because I feel like the brain wouldn't really have a squished capillary.
Right, but if you have anything that obstructs a certain capillary bed in the brain, and you don't have an alternative route of the blood to irrigate those cells that are supposed to get the blood from the original bed, then you can have brain damage. And yeah, without alternative, alternate routes in the brain, that could be disaster. Yeah. So very, very important for keeping brain function. Okay. All right, some places you're not going to find it, okay? Retina, kidneys, spleen, you're not going to find those anastomosis, okay? Okay, so let's see here. So here's an idea here of your anastomosis shunt, okay? And this is in particular what happens in the case of your guts. So this is called mesenteric capillary circulation, right? And just from experience, we're almost done with this. Okay, just by experience, you know that certain situations that you are feeling nervous, for example, you're not going to be digesting your food very well. Well, because when you get nervous, you get sympathetic type of nervous stimulation. Sympathetic stimulation is going to contract those smooth muscles. And first thing it's going to do is going to increase your blood pressure, right? Second thing it's going to do is going to stop the circulation into areas that are not vital. So if you're very worried about, I don't know, running from a predator or digesting your food, your body will prefer to run from the predator and forget about digesting the food. So this is the part that you shift the blood from areas of high absorption, that is in your gut, right, to other areas that you will need to use the blood in a more save your life type of way, okay? So this, when you look at the system that you see in the gut, you really see this anastomosis, that you have this central arteriole that is called the meta-arteriole, okay? This meta-arteriole is going to connect one side to the other. Okay. All right, so this is the last picture showing that, again, if you stretch out in series all of the blood vessels of the body, you would have about 62,000 miles of stretched vessels, and he says that you would also be dead, but that's besides the point. That was the last